Wasn't there? Okay, thank you. All right. Good morning. Since I was unable to take the class yesterday, I'll be taking both hours now. And it is a little bit of an intensive topic, so I'll do it in small bits twice. We'll take a break in the middle. We will be looking at systemic anatomy, the next system which we are going to talk about. Is the nervous system. So I've labeled it number 4 here. What we have done so far is the musculoskeletal system with all its components, bones, muscles, joints and cartilage. Then we looked at circulatory system. We talked about arteries and veins and capillaries and in between channels, their architecture and their function and their dynamic adaptability to the flow of blood as a tissue across all parts of the body. The third one was the hemolymphoid system, which I didn't touch upon because blood as a tissue is mostly dealt with by the physiology department in great detail. But there is a whole bunch of lymphoid tissue and organs as well, which we will do in histology. So remember that there is a separate system, system number 3, which we have to look at or we have to put together from what we learned in physiology and what we will learn in histology. I am not giving you a systemic overview, partly because I want you to do it as an assignment for yourself, where you will try to get together the components. If you can find a general anatomy class, a textbook where this is outlined so that you have a brief idea of how blood and lymph work together as a separate mobile fluid tissue. It's completely different from all the other solid tissues that we have, and therefore as a system which has a capacity to move in and out of intravascular channels. So vasculature is the fixed pipes and hemolymphoid is the movable content. That's a different way of looking at it. That's why physiology deals with it in a much more precise way. But you will be coming across blood as a very congealed tissue in your dissections in anatomy, but we don't look at it as a thing to study in the subject of anatomy. Same with lymph. But we will look at lymphoid organs, where they are and what they do and what differentiates them as per their function, as per their age related how shall we say handling of your body's immunity as you progress from fetus to old person. So we'll leave that, we'll park that for the time being, and we will go into the fourth system which we're talking about today, which is the nervous system. Now, as you already know, this is all very basic human biology. The nervous system is divided conceptually. Please remember that all of it works together. In fact, you can't even separate nervous system from everything else. It is so intimately in control of who you think you are and who you physically, physiologically are. But for the sake of study, it's always useful to divide it into subsets. So we call that central nervous system, CNS, peripheral nervous system, PNS, and autonomic nervous system. So central nervous system is the brain and the spinal cord housed within a very secure bony cavity, the skull, the cranial cavity, and the vertebral column. It is of a highest evolution nature and therefore of a highest sophistication and complexity. From it, an extension spreads out, which we call the peripheral nervous system, which has exited these bony protected areas and is therefore now out in body spaces and in tissue planes and in how should we put it, physically diverse locations, but it is branching out or streaming out from this central axis, which we call the central nervous system. And once it is out there, a large, in fact, by definition, it is serving the body wall. So it is serving what forms your facial skeleton, the neck region, the torso, which consists of your thoraco-abdominal cavities, including the pelvic cavity. So the central part of your body architecture is what we are calling the body wall, and appendages which have grown out from it are your two upper limbs and your two lower limbs. So the peripheral nervous system also serves these four extensions, everything that forms them, and serves the functions of those four limbs that you have. The autonomic nervous system, on the other hand, parallel to and dovetailed with and supplementary to the peripheral nervous system, does the same service of sensation and motor control for the viscera and the organs and the unconscious parts, functions of your metabolic activities which are happening with your digestive system, your urinary system, your genital system, that is your reproductive system, your endocrine system, and so on and so forth. So visceral organ functions broadly are all served by the autonomic nervous system. The word itself means it is autonomic, it is not under your conscious control. So though everything is finally wired to the central nervous system, the differentiation between PNS and ANS is the PNS is still in a conscious connection state. Your forebrain, your brain and spinal cord can send voluntary signals to give orders to the peripheral nervous system. They do that. That is how they operate the body wall and the limbs. But how your stomach is digesting, how your heart is beating, how your lungs are expanding is not under your voluntary control, unless you count yogis who have apparently been able to bring it under control. But that's material for another class, for a more philosophic class. We won't go into that right now. For all practical purposes, PNS is an extension of the CNS control, voluntary control parameters, whereas autonomic nervous system has been taken over by one part of the CNS called the hypothalamus, which is the master controller below the level of cortical consciousness. We'll come to that in greater detail when we do the nervous tissue in detail. Right now, let's look at it from a systemic format. The basis of all your nervous system is a very, very, very specialized cell called a neuron. We are all aware of this. But what we understand when we use the word neuron is very different, because there are several types of them. For example, this is what we imagine when we see or think or talk about a typical neuron. It will have a body, a cell body, right, which is somewhat spheroid, ovaloid, ellipsoid in shape. It will have a nucleus inside that, and in that there will be a nucleolus, which is usually quite prominent. I think parallelly you are doing cell structures and organelles with biochemistry, so you can correlate now that we're looking at a very, very specialized cell, but it will have similar organelles as anybody else. Some organelles will be more pronounced because they have much more specific functions, much more exhibited functions than other cells. So for a neuron, for a neuronal cell, the nucleolus becomes very prominent, and it is clearly visible when you look at it in a microscopic view. So this soma, or the cell body, is basically ellipsoid shape, and it has many, we're only showing some 10 or 12 here, but it has a few hundreds, sometimes a few thousands of extensions from the cell body, which are very thin dimension processes or extensions, very little bit of cytoplasm going into these very, very, very long extensions housed by the same neurolemma, the same cytoplasmic membrane of the neuron. And those are called, most of them are cellular processes called dendrites. It's a specific name we give to cellular process extensions of neurons only. We don't use this word dendrite anywhere else. So because it looks like a multiple branching situation, where the singular extension, if you see, it branches again and then branches again. So we call it a dendritic tree. And if you keep on tracing it somewhere to the end of it, it will go and end in a situation where it is forming a kind of a joint usually with either another neuron or with another cell, which will do something because the neuron passes a signal to it. Right? If it's a muscle cell, it will contract. If it is another gland cell, it will secrete. If it is something else, it will sense, if it's a special sensory organ. So there are multiple functions which can happen with the next cell with which these dendrites have connections. But what we are also seeing is incoming connections from the next adjacent neighboring bunch of neurons, and they come and they end in little button-like expansions right on the surface of the cell body. So though here we've cut it away to show that as if it's an open view, these endings are not happening in the cytoplasm. Please remember, they're happening on the membranous, that is the cytoplasmic membrane of the cell body, or the soma, is where these endings are happening. This is on the soma, this is on the dendrite, this could be anywhere on the cytoplasmic membrane of this, which is covering a huge surface area. When you take any one neuron, the amount of cytoplasmic membrane it has, because of these hundreds of branches, is huge. So we have a soma, or a cell body, which contains a nucleus and cytoplasmic organelles. All the organelles will be here. You can see some endoplasmic reticulum, some mitochondria, some Golgi apparatus, whatever, all kinds of stuff is there. The dendritic tree will have the synaptic contacts which end in it, and there is a third item called the axon. So there will be one wider dimension extension, wider than the dendrites, right? Comparatively wider than any of the dendrites will be one, just one extension of the cytoplasm into a long cylindrical process which is called an axon. And where it starts is a little expanded piece of cytoplasmic material which is called the axon hillock. And the proximal part of the axon is its broadest part, and then it tends to get a little narrower. Now this is the general architecture of a neuron from almost anywhere, particularly those from the CNS. PNS and ANS neurons tend to be a little more diverse, in the sense there will be 5 or 6 types of them depending on what they do. The broad differentiation is between motor neurons and sensory neurons. We will come to that in a second. So in this picture, it's basically showing you all the components of any one cell, and in that we know that there will be a cell body which we call the soma. There will be several extensions called dendrites. There will be a singular bigger extension called an axon, which will start at a place called the axon hillock, or rather wherever the axon seems to be starting from the soma is called the axon hillock. Inside the soma will be the nucleus. Always the nucleus is never in any of the processes, never. It's always housed within the soma, which is the biggest. The common sense is because there's no other place in the neuron which has enough diameter to house a nucleus, right? The only place a nucleus will fit is in the soma, right? And in that it will have its nucleolus. It will have inside the nucleus you will have all the DNA, the chromosomes, nuclear material, nucleoplasm, etc. Some amount of RNA, and so on and so forth. The synapses, the word is a nerve-to-nerve contact, or a nerve-to-nerve junction, where some communication will happen. Usually it will be of a biochemical nature. Some chemical material will be released from one of the cells to the other at that junction, which we call a synapse. Like I said, we'll differentiate between motor neurons and sensory neurons. So in motor neurons, depending on what kind of motor function is happening with it, which means that motor function, motor means movement, right? Your car's motor is that which brings about the movement of the car. Okay? You've done this in physics. So motor as a root word is always talking about movement. So a motor neuron is that which brings about some kind of movement. And most of them, if we do not otherwise use any other adjective or any other qualification, we mean voluntary movement. Alright? So a motor neuron, the dominant type, which is there particularly to bring about locomotion movements in your body and movements of joints, movements at joints of limb parts against each other. This is having that typical picture which we saw in the previous slide, where you have a big soma, you have a big nucleus in it, you have lots of dendrites, and you have one long axon. Now in this particular one, this doesn't seem to be the biggest branch. This looks like bigger than the others. Don't worry about it. Always the axon will be the longest branch. It will usually be the longest of all the cytoplasmic processes of a neural cell. So inside the soma, the extra thing, I think again your biochemistry people will tell you in great detail, there will be some visible small dense bodies called Nissel's bodies, always seen only in the soma, which are usually missing from that place called the axon hillock. They're not seen there anymore. What they are, what they do, I'm sure they'll tell you in biochemistry. I'm not going into it right now. I will check whether you remember this when we come to neural histology, which will be much later. So in a large motor neuron, you'll have one dominant long, very, very big axon, and it will always end in a muscle because its function is to bring about movement. And the only tissues we have which have the capability, the capacity to bring about movement by contraction is muscular tissue. So the kind of muscle which will cause movement which we can command is striated muscle or striped, right, or voluntary muscle. So striated or skeletal muscle receives the neuronal endings from these motor neurons, and that situation is called a neuromuscular junction. Now that will be taught to you again in very high detail by physiology people, because they will talk about the muscle nerve activity in physiology in great detail, and which is applicable as in principle to everything, every kind of muscle nerve interaction that we call. So that NMJ, neuromuscular junction, the specifics of it, the details of it, they will take up. What we are looking at now is the types of neurons that we have in the whole system, which we call the CNS. So the motor neuron is of this nature, this kind of shape and size and architecture, and functionally the function of it determines how its parts are going to end up. So this one's end part is going to be on striated or skeletal muscle. On the other hand, you need some movement happening with your viscera also. So if viscera are going to have to bring about some movement, such as digestive peristalsis or whatever, you need another muscle to operate it, and the ones which are found in viscera are smooth muscle or involuntary muscle or unstriated muscle. They were those spindle-shaped ones we saw in the last few classes, which had a central nucleus. They didn't have these long cylindrical syncytial multinucleate structure. Rather, they had these bunches of spindle-shaped cells closely opposed to each other. You remember they had a lot of intercellular junctions, and we said when they're stimulated by a nerve, the waveform of contraction is shared between a whole bunch of spindle cells. This is extremely important to bring about the kind of coordinated squeezing that's required for your gut. So the nerve which supplies it is not going to be like your typical motor neuron. It is also going to have a soma. It's going to have a nucleus and nucleolus. I hope you can see that. It's going to have a very rich dendritic tree as expected. It's going to have an axon. And suddenly, it's going to pass on that message to another one, another autonomic neuron. So the neuron which is going to be receiving this signal also has a cell body or soma, also has a dendritic tree, also has an axon, and that is going to end up on a smooth muscle, for example in the intestine. So because a nerve to nerve junction has happened here, we said that is called a synapse. So these two neurons which are participating in this control of that smooth muscle of the viscera will be presynaptic or postsynaptic, right? The one which is bringing in the initial order, and then the one which is relaying that order to the actual muscle which is going to contract. So they are named as presynaptic and postsynaptic autonomic neuron. Just remember this terminology, because we'll be keeping on coming back to it when we actually see it in dissection. We'll say, okay, we'll pick up one neuron, we'll pick up something, and we'll say, okay, what could this be? It's probably a nerve, because of its location and because of where we have done the dissection. What do we expect this nerve is doing, or where is it going and what is it going to do? We can trace it in dissection. Where is it coming from and where is it going? but we must remember the plan. We must remember the way it is put together in the plan of the human body for it to be able to cause its function. Always function will underlie the structure. So the motor neuron, what we call motor neuron, could be motor in a voluntary sense where it is controlling skeletal muscle and it is of this large motor neuron type, or it could be motor in a autonomic sense where it is controlling the mostly the visceral and organ smooth muscle, and there usually it has at least two neurons in a relay system which is called a presynaptic and a postsynaptic neuron. Once a neuron ends up on a muscle, the way it ends is a complicated structure called a neuromuscular spindle. So this is supposed to show you the skeletal muscle with the striations, right? So on this, if you take any one skeletal muscle or voluntary muscle fiber, you see we already saw it is cylindrical and very, very, very long, and it is a syncytium because all the nuclei of that myoplasm have been shared, have been pushed to the periphery to make place for those contractile filaments filling up the center of the long fibers. So this long structure is the skeletal muscle fiber, right? Muscular fiber, which is in parallel bundles with each other. It will receive a incoming motor neuron nerve ending, right? Which means axon is dividing into so many, a bunch of branches, each of which is going to end up on any one skeletal muscle ending. So when you examine that ending in high detail, you see that the end point of that neural structure is expanding out, and it seems to be containing some blue and some red fibers, which means let me see, probably some of it is sensory and some of it is motor. My guess is that the blue ones are motor. Usually those are the colors we employ, but it could be different in different atlases and different textbooks. Please check and read. Okay, so what we are saying is we're using a word here called efferent, and we're using a word here called afferent. So afferent means incoming, it is coming inward. In this case, this whole thing has the nerve cell body is probably somewhere in the spinal cord, in the CNS, either in the brain or in the spinal cord, and the fibers are going out from it. But in the same bundle, if you look at it from outside, we don't know whether it's incoming or outgoing. But in the same bundle, there are some branches which are bringing in sensation, and there are other branches which are taking away orders, which are taking away instruction. So here we use the word afferent as if afferent means incoming. So these afferent ones, I think, right? The word is afferent. Can you see the blue ones? They are afferent to the cell body. Okay, they are taking away information towards the cell body. So one way to remember it is equal to sensory. What information, any information which you take to a nervous system is sensory in nature. It need not be conscious sensation, but it is bringing in some information which will be interpreted in some form of sense and then acted upon, and then the action will go, okay, now do this. Oh, this is the condition in which you are. All right, now I want you to do this. Right? So then that means the color coding here is opposite to what I said. I'm just making sure you're awake. Please be awake. So afferent is where you have a bunch of little network like endings, very small, where primary annulospiral endings of a group of afferent fibers are seen, where inside the muscle spindle, inside the muscular fiber, there is an area which seems to be a little expanded, little bulbous, little spindle shaped. On that, we are seeing that there is a whole bunch of afferent information which is going away from that in this bundle, right? But another bunch of information is coming in as red, which are efferents. This one is called a gamma efferent fiber, which may have a trail ending or a plate ending. Okay, so it may end like a little network with a loose pinpointed tip, or it may end in a small plate-like shape, and it may end on the spindle. This is just the look of it when you look at it under high power microscopy. The functions of it will be slightly different depending on that. So you have a plate ending of a beta efferent fiber. So which means your efferent fibers are further being grouped according to their diameter and according to the function that they subserve. So right now we got two words from this diagram called gamma and beta. Usually if there's a beta, there must be an alpha. Let's see, in this labeling, is there any alpha? I can't see an alpha. Okay, so this one says this is a secondary flower spray ending of a group 2 afferent fiber. So blue is a separate category where it is taking away some sort of information from the neuromuscular spindle. The word secondary has been used, which means there must be something primary. So primary plus secondary together are afferents, blues, which are taking information away. They seem to be taking it away from two different sources. One is this expanded spindle shaped part of the muscle fiber. This bit is been labeled as a nuclear chain fiber, and this as a nuclear bag fiber. I'm not going into the details of this. I'm not even sure how much of this you need to know. We usually make this a PG question. But in your book, however deep it has gone, please look at it. Just look at the labelings in your diagrams. It's probably much more important in physiology. In anatomy, we won't be showing you this. This is at the electron microscopic level. So we will only be showing you simple muscles ending at about 100x, maximum at about 400x magnification. So how much structure you can discern at that magnification, everything about it, I will expect you to know and to answer in an exam. This is actually a little more deep detail, just to let you know, just to bring home to you that there is a concept called a neuromuscular spindle, in which there is afferent information which is being subserved by primary and secondary endings of afferent fibers, and they're taking away information towards the CNS. Then there is a whole bunch of efferents which are coming from the CNS to this neuromuscular junction, where they will give orders or they will release neurotransmitters which will cause some action. For a muscle, the action is contraction. So here we are saying there is a gamma efferent, and here we are saying there is a beta efferent fibers. The difference seems to be that the beta ones are going towards the tips of this spindle shaped structure, whereas the gamma ones, the trail endings and the plate endings, are ending more on the thicker part of the spindle of these fibers. In between, with these spindles, with these nerve endings wrapping around it, there is one green material which is surrounding this whole bunch together like a wrap, which we can call a capsule, and therefore there is a subcapsular space. And in that capsule, if you look at it even more carefully, there will be an internal capsule and there will be an external capsule. So somebody has fine dissected it, laid it out, and discerned all these spaces, all these components, given each one a name, and then studied what each of them must be doing. And imagine, to be able to make a muscle contract, it has to firstly be in a live state. You can't do it in dead cadaveric dissection. So some exquisite design of research must have been done to be able to get this information from any one nerve muscle interaction. And this must have been done many times to establish it as a scientific fact. I chose to go into this much detail, though you don't need it, to bring home to you the rigor, the precision with which we have to design our experiments to be able to get this much information. Series of experiments revealing little information at a time. Then we put all the information together and come up with the concept, oh, this is how it works, right? It is quite fabulous. And you are young scientific minds. I want you to already think in terms of applying yourself to, oh, we know this much, but this we don't know. What can I do to get to add to this knowledge for the whole medical fraternity and in the service of your patient? All right. So just the concept of what is a neuromuscular spindle. Please go through it in your book or whenever you're doing your self-directed study. And I wanted to familiarize you with this word of afferent fibers, efferent fibers, plate endings, bag endings, flower spray endings, alpha, beta, gamma, all these names which have come. They're not extremely important for each of you to know, but at least you should know there is something called a neuromuscular spindle and there are incoming and outgoing fibers. Now you must be thinking a muscle has to contract and the nerve has to make it contract. So what sensation is happening? Why will muscles feel anything? Muscles only act. But the CNS at all times needs information of how much action has been executed. I asked you to contract 10 grams worth. Did you actually contract 10 grams worth? So in a dynamic way, every microsecond, the state of contraction, the state of action of that target cell is being relayed back to the CNS. And depending on that information, the next instruction will come. And so it's a loop, an information action loop, which is quite super fast. It's happening in microseconds. And that's why you're able to do such controlled activity, particularly with your fine muscles around your oral area, with your fingertips, with your eyelids, with movements of your eyeballs. All of these are voluntary actions which you're able to bring about with exquisite precision, particularly if it is a trained maneuver which you have practiced many times. That lays down these neuronal networks. It establishes and makes strong those networks which allow you to do precise activities without even thinking about it, right? I don't even know what is happening or what is the position of my pencil, but I've done it a few thousand times in my life, which is why I won't drop it. I will put just enough pressure, just enough movement, just enough power in a dynamic way, because one part of my brain is decided to show it to you on camera. The other part of my brain is concentrating on what it is that I'm trying to teach you. So this multiple complexity is only possible because we have so much architecture, material invested. We've given you a few thousand fibers over there. How many of them you employ at any one given point of time depends on the basic individual organism, the individual person who is doing it, the amount of times they've done it, the sophistication of the neuronal circuitry. And the executive portion, the one which carries out the task, is the neuromuscular spindle at each and every neuromuscular junction. If you look at it, if you actually look at it with a special nerve stain, you will see that mostly we use stains which are lipophilic, because nerves are very rich in lipid, in fatty material. So the stain we usually use is brownish black in color. So the nerves stand out like black structures against the background of whatever other tissue which won't take up that particular stain. So it looks like, as described, as shown in the drawings and the paintings, like a knobby, rounded button-like endings of a tree of branches arising from the end of the nerve, and where it lands on the muscle myolemma, the cytoplasmic membrane of the muscle cell, will be a small ovoid, rounded, oval ellipsoid kind of a structure, which is called a motor end plate. This is different from a synapse, which is a nerve-to-nerve connection. A motor end plate is a motor neuron to a skeletal muscle connection, right? So when it ends up here, you see that this is the yellow and the blue-green is the nerve ending, and all the red-orange is the muscle, right? So the nerve comes, and inside of it, you can see that's the axon, the blue-green. The yellow is the wrapping. It is the myelin sheath, which I said is lipid-rich, which is a very important insulation material which has been provided to separate all the traffic which is happening in one nerve and its branches from the next adjacent nerve and its branches. The body is so economical that it packs a whole bunch of structures into very tight spaces. So when they're so tightly abutting on each other, the instructions in one must not get confused with the instructions in the other, which is why they have to be insulated from each other. So this myelin sheath is a lipid material, again very sophisticated biochemistry, which makes sure that impulses in one neuron ending are not getting at all communicated to instructions in the other neuron ending. Both of them can function independently of each other, even if they are running in very tight juxtaposition within the same wrapping in a nerve bundle. So the motor axon here is dividing into its final branches, and it is landing on the surface of the muscle cell, right? Where it is landing on the surface of the muscle cell, there seems to be a zigzag kind of a situation. That means the muscle myolemma, cytoplasmic membrane, is thrown into a bunch of folds, increasing the surface area essentially. And inside the ending of the axon, there seems to be a whole bunch of small dot 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 structures, which are nothing but synaptic vesicles. That means, like I said, we use the word synapse only mostly, almost entirely, for nerve to nerve endings. But here also we're using the word here, but you must remember this is the motor end plate we are talking about. And the muscle part of this junction, which is with this expanded surface area, is called the muscle sole plate, S-O-L-E, like the sole of your foot, right? It's because this thing is coming in, landing on it. And the nerve part of it is called the motor end plate, which has the synaptic vesicles. So the motor end plate is the motor axon ending, and the muscle sole plate is the muscle side receiving surface with a very corrugated shape, where not the nerve side is not corrugated, it's very smooth, but the muscle side is a very corrugated shape, increasing the surface area of the contact, of the congruence between the ending of the nerve and the receiving part of the muscle. So this whole thing has again sophisticated geometry and allows communication usually from the end. In this case, since it's a motor end plate, all the information is going from nerve to muscle, right? And the release of that chemistry will stimulate this surface membrane of the muscle and depolarize it, sending off a wave form towards those actin myosin filaments and making them slide against each other and bring about a contraction. Should we stop here now? How much time has passed? I think I'll give you a little bit of a break and then we'll go to sensory neurons. I think that makes sense. Well, let's see. Okay, I'll finish a little bit more and then we'll take a break. So we were talking about motor neurons till now. The general plan of it, the fact that there are several types, out of which the major ones we looked at were for the ones which are ending in neuromuscular junctions and the ones which are ending in as part of a presynaptic postsynaptic autonomic plan for the visceral muscle movement. The reverse, the opposite, equal and opposite, another huge few million neurons that we have, are for taking away sensation from surfaces which are sensory in nature. So here again, the plan is the same. You have to have a cell body, and you have to have cell processes, a whole bunch of cell processes. Now the logic is, if I'm going to be a motor cell, my axon is going to do the ordering, right? So my dendrites will receive whatever information or whatever instruction from somewhere, or my soma will generate the instruction and my axon will take it away and go and distribute it to the whole bunch of things which are going to execute that order. That is a plan of a motor neuron. So whether you have a big soma, you have a rich dendritic tree, or you are basically only soma with one axon, all of that is possible with the motor side. Or you could have 2-3 cell relays. One nerve talks to another nerve, talks to another nerve, finally talks to the muscle. It doesn't matter. The point is that is the way the message, the neural message is relayed is in a biochemical flow of molecules within the neuroplasm, always away from the soma into the axon and then out. A sensory neuron is the one where the dominant thing it is doing is bringing in stuff, information, electrical information, sometimes, mostly, sometimes very clearly through a ionic flow in the neuroplasm. But the dominating part of this neuron would be the dendritic tree. It could be a single dendrite, it could be 2 dendrites, it could be 1,000 dendrites. All of them will be dominating. The axon part, which takes away the information, will be usually, it's always singular, and it may not be very dominant. It may just be relaying it to the next, and then to the next, and then recording it in your conscious or unconscious parts of your sensory cortex. So a sensory neuron will again have a soma, and will have a dendritic tree, and will have an axon and an axonal tree. It will be called bipolar if it has one main incoming afferent cellular process and one main outgoing efferent cellular process. Now this is different from what we saw in multipolar neuron, where all the dendritic trees were landing directly on they were continuities, or they looked like branches from the soma itself. But the bipolar neuron is a very common kind of sensory neuron you have, where the incoming information, this tree will be much richer, it will be bringing in the information, but its connection to the main cell body will be singular, and its efferent connection to the next cell to which it is passing the information will also be singular. So there are only 2 poles. The soma cell body has only 2 areas where cellular processes are seen to be attached, that's why it's called bipolar. Then you have what looks like a cell body with only one process. That would make it unipolar. So neurons can be unipolar, bipolar, or multipolar. The classic motor one we saw is a multipolar neuron, but the unipolar as a kind of neuron actually doesn't exist in the human species. What we have is a pseudo unipolar. We call it pseudo because in this bundle, in what looks like a singular process, actually there are 2 ways of traffic. There will be sensory things coming in from one side, which will be its dendritic component, and there will be instruction things going away in its branch process, which will be the axonal part. So in this case, there are sensory nerve endings in the skin. So the dendritic tree is forming one singular channel, which is bringing it in, which will bring it to the cell body, and information from this will go away in the axon in the opposite direction. So actually this, what looks like a single process, is doing 2 things, which is why we call it a pseudo unipolar neuron. We'll see quite a few of them in the sensory part of your nervous system. Let's look at all the kinds of nerve endings we have, and then we'll pause, we'll take a break, and we'll come back to the rest of the nervous system. So this is a single picture, but I'm sure your textbooks or your reference books will have very nice illustrations. Please go and refer to them. This now I will just give you a bird's view of all the kinds of sensory endings that we have. So we said the dendrites are bringing information. So the tips of the dendrites must be the areas where the information is coming in. So these tips of the dendrites can be loosely in the sense that they'll be just ending blindly in a sharp tip of cytoplasmic membrane, hardly containing any cytoplasm, and just lying there in the tissue. Then they're called free endings, right? So this is the nerve bundle, right? So a whole bunch of dendrites here, a whole bunch of dendrites here, and this is going to be taking it away to wherever the main nerve is. The soma is nowhere here. The soma will be somewhere inside the CNS, brain or spinal cord. Only the peripheral processes, the axons and the dendrites, will be seen by us, and that too only in big thick bundles. We won't see single nerve fiber endings almost anywhere. Now this is a section of skin. Okay, you can see this is the epidermis. I think you have studied this enough number of times. And this is the dermis. So the epidermis part, at its junction with the dermis, seems to have a whole bunch of nerve endings, which is common sense because your skin is one of the most sensitive parts of your body. It's an extremely important and the biggest sensory organ that you have. For that sensation to happen, several kinds of sensory endings have been provided, of which the first one I'm talking about is free endings. So these so-called free endings are tips, beginnings of dendritic processes which are just lying there, usually in a dermis part of the skin. They could be again of three types depending on what sensation it is that they are tuned for. If they are tuned for mechanical pressure, they can sense a mechanical disturbance in their environment. They're called rapidly adapting mechanoceptors. Okay, another kind of them which doesn't look very different are called thermoceptors. They are able to pick up sensation of temperature from their environment. Then a third type which are called nociceptors. Noci is pain. They are able to pick up enough derangement in the environment which is interpreted as detrimental to the tissue or detrimental to the organism, right? And that sets off a sensation which we recognize in the central part as pain. In the periphery, those specialized nerve endings have been provided. All three of these, mechanoceptors, thermoceptors and nociceptors, are free endings. They don't have any particular shape or size or anything. They're generally located all over the dermis part of your organ called the skin. Actually, of the system, the integumentary system, which includes the organ called the skin. So these three types, all three of them together, are free endings. Apart from that, there is another kind of ending which is looking a little ellipsoid in shape, and it is particularly seeming to be located at the epidermodermal junction, only at the baseline of where the purple line is what separates the epidermis from the dermis. That's where you're seeing more of these purplish brownish in this painting. Okay, in life they're not purple, they're not any particular color. This is a HNE stained section of skin, and in this particular picture they've painted this yellow. It doesn't look like that. This is just a painting. Please don't go by the colors, just to make it interesting for you. Imagine all of this is black, white and gray, and you see that the shape and size of this kind of ending, sensory ending, is a little dense looking and a little ovoid. And if you look at it under high power, this is what it looks like. And these are called Meissner's corpuscles. I'm sorry, my labeling is, I didn't do separate labeling. Meissner. This is a German scientist who first named them, who first found them, described their architecture, figured out what they do, and named them. And he used the word, or we use the word, corpuscle to describe it. We could just call it Meissner's ending or Meissner's nerve ending or something like that. But we use the word corpuscle. It has stuck and it is continuing. Corpuscle. Corpus means body. Corpuscle is a small body. So a small ovoid or ellipsoid body, inside which there seems to be a spirally arranged bunch of nerve endings. Same nerve ending but in a spiral arrangement. And inside a kind of a sheath, there's an extra bit of tissue available which is surrounding it. And this whole body together seems to be sensitive, particularly to pressure. So it's also a mechanoceptor, and it's called a Meissner's corpuscle. This is also a mechanoceptor, but this is a free nerve ending, and it is a rapidly adaptive field mechanoceptor. Okay, this is also rapidly adapting, but it's just called a simple mechanoceptor free ending. You remember, a Meissner's corpuscle is for mechanoception. Okay, mechanoreception. Then you have another kind of a ending which is not so superficial at all. It seems to be embedded in the fat layer of your dermis, right? In the beneath the skin layer of fat, superficial facial fat that you have, a rather large size receptor compared to all these little very tiny ones. This seems to be very big in size, right? It's a huge one. It seems to have a very specific architecture. So when you look at it expanded, you see that the nerve ending is expanded into a bulbous thing. It seems to have some layers, fibrous layers or something laid down in concentric ovoid or ellipsoid shape. And at the core of it, the nerve ending, this is its Schwann's cell, its myelin sheath. Then there is a bare part, and then there is a different tissue. It's as if the tip of the nerve fiber is embedded in some material, and that is surrounded by some other material, and that has got another wrapping around it, and it's rather large in size. And this whole thing is called a Pacinian corpuscle. This is an Italian scientist. We honored him by naming it Pacinian, Pacinian corpuscle. Again, we are using the word corpuscle. So a Meissner's corpuscle, which is a mechanoceptor, and a Pacinian corpuscle, it's different because it seems to be lamellated in nature and much larger in size. So a Pacinian corpuscle is a pressure receptor particularly, therefore it is more deeply placed. It is not superficially placed. Sufficient amount of pressure has to be generated for it to be stimulated and for it to send its signal, and it is deeply placed in the dermis or in the superficial fatty layer of fascia, and it is surrounded by blood vessels and so many other things are happening. It is a bigger body, it has a good blood supply. Okay, so you remember it as a lamellated Pacinian corpuscle. So the differentiation between Pacinian and everybody else is it has lamellae, a lamellar structure. Then you have the ending near some fibrous structures of the dermis. So already the dermis is containing many fibers being laid down, right? That is your collagen, elastin fibers, which are forming the bulk, the material of your subepidermal region, that is the dermis itself, and the hypodermis, which is the subcutaneous tissue. So in that area, you're seeing something called type 2 slowly adapting mechanoceptors, or Ruffini endings, also named after an Italian, Ruffini, Ruffini, where the shape is that the nerve ending comes, its myelin sheath stops, but it expands into a kind of a spindle shape and wraps around the fiber bundle, getting in between the individual fibers, right? And around of that, so it's getting into the fibrous sheath of a fiber bundle in that dermal territory, and it is wrapping itself, its network, its tree, it's wrapping itself around that. So that whole package together is called a Ruffini ending, and its nature is it is slowly adapting, as opposed to this rapidly adapting and this rapidly adapting. This one is a slow adapting mechanoceptor. It is required for us. So remember, there is one more kind of ending which has a different shape, which is called a Ruffini ending. And then we have a commonly seen one, which is again exactly at the epidermal junction, also looks ovoid or hemispherical, half spherical shape, and particularly found closely crowded where the epidermal pegs are. The base layer of the epidermis locks into the dermis in the form of rete pegs. We will look at it when we look at the structure of skin. But this is so that the skin, two layers, that is epidermis and dermis, do not separate easily from each other when there is a shearing force applied on the surface. So wherever you have epidermal pegs going in and interlocking with dermal pegs is where you have more number of these endings, which are called Merkel, M-E-R. It's again a German word. I think it's spelled C-K-E-L. In this book it's spelled M-E-R-K-E-L. I think that's more correct. Merkel cell ending, which is also a mechanoceptor. There you can see that the nerve ending is typical, the way a nerve is supposed to end. This is how we saw the dendrites are ending in these little button-shaped endings. And its sheath also has ended at that epidermal junction. And these are epidermal cells all around. But there is a separate one thing, which is the actual sensory ending of that nerve, which is having some vesicles or some chemistry over here, and a how shall we put it, hemispherical dome shape with some extended branches which are abutting on each of these cells. So if these cells are going to be mechanically disturbed, this will get stimulated and the nerve signal will go. This is a Merkel cell ending, and it is also a slowly adapting type. So Merkel and Ruffini are slowly adapting. Pacinian is lamellated, right? Free endings could be mechanoceptors, thermoceptors, or nociceptors. And one type of rapidly adapting mechanoceptor, which is not a free ending, is a Meissner's corpuscle. It is a bigger size and it is available mostly at the most superficial parts of the dermis of the skin. So these 6 types, at least with the names, if you know, that's good. These sensory endings are the ones which are bringing in all the sensory information from the external environment to us, and all that information is being passed continuously into the sensory part of your system. Much of it, since it is coming from the body wall, is of a conscious sensation nature. Just like your motor action is either voluntary or involuntary, like that your sensory action is either conscious or unconscious, right? So what is happening, whether your stomach is expanded now or contracted, you don't know, you cannot tell, because that's not in your conscious sensation. But the information is continuously being passed to your CNS. Whether or not it will be related to your sensory cortex is a function of many things. We will come to that when we come to the higher levels of how the CNS operates. A good amount of its attention is given away to lower centers so that lot of function can happen in an unconscious or subconscious state and don't take up the attention of the animal. A comparatively smaller portion is kept in the conscious category, where you are aware of what is happening, and the information coming from the periphery, where you are interfacing with an external environment, is extremely important. You have to be aware of it at all times. So even though the tips of your toes or the tip of your scalp is not an extremely vital organ, information from it is always conscious. It is always available to you, to your sensory conscious cortex. Whereas though your heart is a much more vital organ, or your inner organs which are life or death situations, for that matter the brain itself is such a vital organ, but what state it is in and whether it is under pressure or not, or whether there is a bleed in it or not, is never under your conscious sensation. It's paradoxical. We will look at that from a how does that serve the animal perspective later. Right now, this is the nuts and bolts of your how the motor part and the sensory part of your nervous system as a system are made available, and where they are, how we have named them, what we understand of them, what they do in a generic sense for the whole organism altogether. When we do regional anatomy, we will look at specifics of how these components work together in that particular region to serve the function of that particular region. We'll take a break now, and can we come back in, let's say, 10 minutes, 5-10 minutes? I'll end this session and I'll restart it again in let's say it's 11:05, let's say 11:15, and we will continue with the rest of this.